and if you pardon the background noise, we're also homeschooling kids at the same time, so it's a fun time to be in biocontrol right now for sure. Quick overview. I'm going to give a brief history on how biocontrol kind of came to be. Biocontrol as we know it today, that is. Biological control in theory and practice. Identifying potential biocontrol agents, how that process works. The host specificity testing arena and what that looks like. The approval process. Permits for release. And lastly, how biocontrol and IPM work together. So a brief history about biocontrol of weeds, and I differentiate biocontrol of weeds with regular biocontrol of insects in general, because we don't really work with that, although they do at Palisades, and I'm sure John's going to talk about that after I'm done. The first major success story you probably have heard of, it's been back in the news recently, that's Cactoblastis cactorum on the Opuntia species. That was in Australia in the early 1900s. And you can see on the picture here, the before and after picture. This was in 2 years' time. It was a phenomenal success, and that kind of got the ball rolling when it came to weed biocontrol. Up next in the U.S., that is, we had St. John's wort and Chrysolina hypericea and Quadrigemini were released in California by UC Berkeley, and you can see the before and after pictures of that, and it was also a stunning success. And there's a host of other examples that Harriet Hinds really outlined with some of her colleagues back in a paper published in 2019. In that paper, interestingly, they also did show a majority of the successes that we see today, to include Opuntia, probably would not be allowed today because of the stringent policies we have in place when it comes to governing biocontrol. And that's what I'm going to kind of get into after we go to the next slide here. And I don't know why this is not working. There we go. So just to make sure we're on the same page, biocontrol in theory and practice. There's a lot of different definitions of biocontrol, and if we're going to talk about it, we might as well be talking in the same pretense. Biocontrol, classical biocontrol is kind of what we work with when it comes to weed biocontrol, and that's using a non-native insect or pathogen to control another non-native. A lot of folks don't understand these plants that we have that are huge problems did not originate in the US. They are not native to the US. So in order to balance them with their natural enemies, we have to go into that area of the world where they came from and find the natural enemies there. And then it goes through the testing process, which I'm going to outline briefly here in a few minutes. Conservation biocontrol is another form of biocontrol, and that's modifying conditions to favor natural enemy survival. A lot of times we do biocontrol releases, we do releases in areas that are not optimal. So you can actually modify those areas to make them a better candidate site to get biocontrol established. There's augmentative biocontrol, and that's using biocontrol as sort of a biopesticide. One of the nice things about biocontrol agents is they don't have any labels or anything like that associated with them, so you can use them anywhere. That's in contrast to herbicides, where you might not be able to spray within 50 yards of a river or something like that. With insects, you can put them out virtually anywhere as long as you have the proper documentation, and I'll get into that too. And then there's cultural, and I'm going to differentiate cultural biocontrol with biocontrol that I'm going to talk about, and that's using this sort of macro biocontrol agents, I guess I'll call them, goats, sheep, things like that. Those are not necessarily classical biocontrol agents, and we're going to kind of separate those from what we're talking about here today. So diving down into classical biocontrol, what this does is it focuses on simple plant-herbivore interactions. As with anything, there are advantages and disadvantages. The advantages are it is incredibly target specific. We have some biocontrol agents that are not only species specific, but they're biotype species specific within a general species. They are continuous in action. And when you work with big landowners like the BLM or the Forest Service or folks like that, we have limited budgets to kind of go in and do big time control measures, whether it be herbicide or biocontrol. So it's nice to be able to go out to an area that you're not going to be able to get back to, release some biocontrol agents, and know that they're going to be working for you when you're not there to actually do the work year after year. Long-term cost-effective. One of the most recent cost-effective or cost-benefit ratios was done in 2018 by a colleague of mine, Urs Schaffner, and he found that for every dollar you spent with biocontrol, in some instances, it returns $18,000 on the return. That is an incredible cost-benefit ratio. It is gradual in effect, and this can actually be seen as both an advantage and a disadvantage. Speaking from the advantage side, it's gradual in effect because when you go in and do with control measures, oftentimes you're adding an additional disturbance. With biocontrol, you don't necessarily do that. You're not bringing in heavy tools and machineries that might open up another niche for another non-native species to come in and kind of take a foothold. This is gradual in effect, and it gives the natural seed bank a chance to respond once that natural enemy and weed balance is sort of restored. Generally environmentally benign. It is self-dispersing even into difficult terrain. Some of these biocontrol agents can go upwards of 50 miles a year. Some of them are a little bit more labor intensive, and they can only go about 250 meters a year. But for the most part, in a lot of these areas where we have biocontrol established, they're in super steep hillsides or remote areas, and we find them more and more in places where we didn't know we would find them after they've been released. Some disadvantages. It is a protracted time until impact is likely or visible. When most folks work with invasive species, the general consensus is you want to see it curl over and die right before your eyes. It's very gratifying. Think about all the Roundup ads, things like that you see. Biocontrol does not typically do that. You can get outbreak densities where you can see that, but that's not commonplace. Uncertainty over ultimate scale of impact. This can be kind of a pro or a con. It could be really, really impactful in a short amount of time. Salt cedar is a great example. It went from 2 hectares to 400 hectares in 1 year. And it's just the salt cedar leaf beetles just kind of bounded leaps and bounds from there. Or it could take 7 or 8 years before we find any sort of control. And this is sort of the experiment that is biocontrol every single time you engage in a biocontrol release. Uncertain non-target effects of the ecosystem. This is kind of seen with food web interactions, and we can't really with good certainty go through all the food web interactions and project what might be until the biocontrol is released. Uncertainty, or it is irreversible. Once you get a biocontrol agent out there, you are there's a reason why there's a lot of testing on the fore of this whole process, because once it's out there, you're not going to get it back. Not all targets are appropriate targets. This is best seen with the thistle complex. Canada thistle is by far the most requested biocontrol target every single year, and it's also the toughest. I'm sure John's going to talk about some biocontrol for Canada thistle, but for the most part, we don't have a lot of really good answers for Canada thistle because it's in the thistle complex. And we have in the past really done a lot with Canada thistle or just thistle biocontrol, and we found very quickly that it does attack a lot of natives because they're so closely related. We'll not work on every weed in every setting. We have best practices for biocontrol, and a lot of times we go through all the hoops and things like that to make sure we put biocontrol in the best possible situation. And even then, it might not work. On the flip side of that, we might have extra agents after a big collection, and we could go do a big release in an area where we thought it would never take hold, and lo and behold, it does a great job at that site. So every single biocontrol release is sort of a little mini experiment, and that's kind of how we look at biocontrol. We have general rules for what probably will work, but it's not absolutely concrete until we see it out there in the field. I also want to make sure when we talk about biocontrol, we kind of put things in context. Biocontrol is not a silver bullet. Biocontrol is sort of the white flag of control measures when it comes to weed control. Ideal biocontrol results are kind of outlined by this graph right here. You've got the invasive plant that's up above this damage threshold. And if it's up above here, that's where you're getting calls as a land manager or somebody who's working with weeds. So what happens is you release a biocontrol agent right here. And in a couple of years, and notice time is here on the x-axis and population density is on the y-axis. In a couple of years' time, you kind of get to the point where that oscillation sort of bounces below that damage threshold. And over time, you see the desirable vegetation kind of move in and occupy that niche. Now, this could be a couple different things could happen here that could really throw things off. You could have another invasive come in and occupy that niche. That obviously wouldn't be helpful. But ideally, what happens is you get the target weed over here below the damage threshold where it's not a problem anymore. And you've got desirable vegetation that kind of comes in and occupies that niche. So at this point, you can kind of walk away. You can focus on EDRR species or something else that might be more beneficial when it comes to herbicide or other control measures. How does this whole process work? Identifying agents. The first part is you've got to identify the problem. So what we do in the United States is we form consortiums. And in doing so, we kind of pool our resources, because every single biocontrol agent takes anywhere from 10 to 15 years and $1 to $2 million to get going. So any one agency can't really shoulder that responsibility on their own. So foreign exploration of natural enemies occurs once that group has kind of come together, and they talk to their foreign colleagues where these biocontrol agents are available in their native range, and they do a thorough literature survey. They look through all the literature to figure out what the natural enemies are known to science, and also they kind of come up with the gaps that they might find in the field. Then they move it through a climate matching model called Climax. That takes into consideration rainfall, degree days, temperature, moisture, drought, etc. And then they look at season, habitat, soil type. Then they go out and they actually do the field collections, and they look for those things that they've identified through their literature survey. But they also pay special attention to areas, especially in the roots and the root crowns and things like that, that might not be known to science yet, because those are oftentimes the Achilles heel of these invasive plants, and they're also in a lot of situations where we know the least about. So then they'll go through and they do the field collections, and they'll bring those agents that they find, the potential candidate biocontrol agents that is, back to the laboratory for processing. Then they'll run them through an entire rearing process to do two things. Number one, figure out how to rear this in the lab so they can actually conduct some of the tests I'm going to talk about here in the next slide. And number two, rear out all of the parasitoids and other things that might be attacking these candidate biocontrol agents in the native range that might keep it from becoming what it could be stateside if it were to become a biocontrol agent. So you also want to look at identifying agents. And in the past, biocontrol has sort of been a shotgun approach. I'm sure most folks on this webinar are familiar with the knotweed or the knapweed complex. With spotted diffuse, squareos, and meadow knapweed, there are 13 biocontrol agents approved for release on the knapweed complex. And that's all fine and dandy. We've got control with a lot of these biocontrol agents, but what we're kind of getting at now to get away from potential food web interactions that I talked about is sort of finding the Achilles heel of that potential, the target weed, and really focusing our efforts on that. So the site of attack matters, whether that's roots, stems, leaves, flowers, or seeds, or any or all of the above. You also have the destructive stage of the insect itself, which could be the larval stage or the adult stage, ideally both. We have some biocontrol agents that attack the roots and the fine root hairs as larval in their larval stage. And then when they turn into adults, they attack the foliage up top. So that's kind of a double whammy. And that's where we see a lot of really good results with biocontrol. So after you identify potentials, you kind of run them through a risk assessment model. If they're monophagous, they're going to be a great potential biocontrol because the risk to the non-target species is very low. The only problem is those are the hardest ones to find, very few in nature. You can get away with oligophagous biocontrol agents if you have a very, very unique genus. For example, Chondrilla gensea, rush skeleton weed. We're looking at oligophagous insects that attack everything in the Chondrilla genus, which isn't a big deal because we don't have anything except for the target weed in the Chondrilla genus in the US. And this is easier to find in nature, and you have medium impact to non-target species, so the testing processes for these are going to be a little bit more stringent. And then you've got polyphagous. Those are going to be grasshoppers, just the things that consume everything. Those are really not great biocontrol agents because the risk to the non-target species is very high. So we kind of pass over polyphagous biocontrol candidates really quickly. Specificity testing. How specific are these biocontrol agents? As I said at the onset, some of them are so specific they are biotype specific. Those are ideal. Also, they could be problematic if biotypes there's some movement between biotypes. Specificity is tested by the researcher both overseas and domestically. What they do is they come up with a plant list that looks at all the potential plants that the biocontrol agent or candidate biocontrol agent could attack. And they run that by a whole list of people that I'm going to talk about here in a second. And in doing so, they figure out where the holes in the plant list might be. And then they test the candidate biocontrol agents. They look at the oviposition of the females, where she lays her eggs. If she can find places to lay her eggs on anything other than that target weed, probably won't be a very good biocontrol agent. Feeding must occur just on that target weed, or it probably gets booted out of the candidate biocontrol agent pool. They go through and they do no choice tests, which means that that insect has to either just consume the target weed, and if it attacks anything else, then it shouldn't even be considered. But if it does nibble on something, they will be moving into a multiple choice arena. Multiple choice arena, think of it as an array. It's like 1 meter by 1 meter. And you have 9 different plants in there. One of them is going to be the target weed that you want it to consume. And then the other plants are going to be those that maybe have an egg laid here or there, maybe some ancillary feeding. And if you put it into one enclosure and find that if given multiple replications over multiple years and in that multiple choice arena, that candidate biocontrol agent just goes after that target weed, it's probably going to be a pretty good biocontrol agent. You examine the results, and after years of data and years of testing, you put that whole thing together and you submit it to a group that I'm going to talk about here in a second, but I thought I'd give you a case study to kind of look at how that process might work. Hoary-crested whitetop is a perennial mustard. It was introduced from Europe in the late 19th century. As you can see from these pictures, it does aggressively invade crops, rangelands, and riparian areas. It reproduces both vegetatively and by seed. The consortium was established for this in 2001, and since then there have been 4 agents that have really shown a lot of potential. It has been funding dependent, but fortunately we're to the point where we're looking pretty good. So these are the areas of the plant for hoary crest that they wanted to get biocontrol established for. Right from the onset, they figured out that Silioides rossii and Milan were not good potential biocontrol agents because they were too oligophagous, which left you with the 4 other biocontrol agents, the weevils and the mite that has recently been approved in 2018, the Ceria drava. So the one that they're really focusing on is Pseudorhynchus cardiaria. It's a petiole weevil. It's been tested for about 15 years. No choice tests happened with 117 test species. 62 of those were native to North America with 4 Fish and Wildlife Service threatened and endangered species. That's a pretty comprehensive test list. Gall formation did occur on 18 test species. This is a big red flag when it comes to testing. Adult emergence occurred on 14 species, 9 of which were North American. Fortunately, none from threatened and endangered. This is another really big red flag. In a series of multiple choice tests, they found that the native Lepidium latipus was consistently attacked, so they decided to go through and do some more testing on this one because it was potentially a problematic native that needed some attention. So in doing so, they exposed Lepidium latipus to the target weed Lepidium and the target weed Lepidium drava at increasing distances. And what they found very quickly was the weevils were not able to find Lepidium latipus much past a half a meter, which showed that if you put them right on top, because these tests inherently, you can see some pictures of those arrays I was talking about. Inherently, these tests are very conservative in nature. A lot of times, these natives do not grow sympatrically with these target weeds. So the insect also has to be able to, in addition to lay eggs, feed, and do all those things, find that the target weed in space and time. And with the data that the researchers did in this case, they found out that it really couldn't. So these are data that they're now adding into some of these petitions that I'm going to talk about right now. So once all the data is done, and I said it takes anywhere from 10 to 15 years, the researcher submits a petition to the Technical Advisory Group, or TAG. TAG was established by the Department of Ag and Department of Interior as an unbiased advisory group. It's composed of 15 governmental agencies. If you can think of every single acronym on the government spectrum, USDA, BLM, Forest Service, Fish and Wildlife Service, they have representatives on the TAG. Reviewers are asked to concentrate on 4 main areas. Taxonomy of both the candidate biocontrol agent and also the plant. A lot of times, the taxonomy of plants does change throughout the course of, in this case, hoary crest has changed twice. So they want to make sure that the right plants are being tested for impact. They look at the test plant list, and they can revise that over time. Anything over 100 is a pretty extensive test plant list because you got to think there's years of replication, and then there's replication of replication per year. So you can imagine these things get pretty unwieldy in a short amount of time. Post-range tests, they want to make sure they test all of the life stages, and the impact to non-native plants is obviously essential to anything that may or may not be moved through the TAG process. So this is a brief outline of what that permitting process looks like. So the pre-release research, this is the bulk of the time. This is that 10 to 15 years and the $1 to $2 million of research I said at the beginning. After they do that, they submit the petition to TAG. TAG can then say, this looks good, and then it goes this way. Or TAG can say, you know what, we need more research, or they can reject it wholesale, and it goes this way. If TAG recommends it, the petitioner applies for a permit from APHIS PPQ. Animal Plant Health Inspection Service, Plant Protection Quarantine, is the governing body for biocontrol. They are the gatekeepers. They're the ones that do sort of the risk assessment equivalent for biocontrol. The APHIS PPQ folks consult with the state plant regulatory official. Every state has one. Typically, they're housed with your respective Department of Agriculture. And then if they say this looks good, they either put together an EIS or an EA. And in so doing, they find that there's a finding of no significant impact or a FONSI. And then you get a permit issued by APHIS PPQ to individual states. There's another chance for that process to kind of go back to the drawing board right here. If the state or the EA process or the EIS process say, this doesn't really look good, we need some more information, that's another check in the system to kind of kick it back for some more research or additional testing. So if all things go well and you get the FONSI, then you can submit an application for permit for release. This is an example. This is my old permit for release, and APHIS typically does permits with a bunch of biocontrol agents on them at once. And they kind of look like this. You kind of list them all, and then you list the state origins where you might get them from. And these are typically the states that I work with that do a lot of collections, and we move biocontrol agents back and forth between those states. This is a 526 form, and this is necessary for a lot of biocontrol agents, but brand new as in the last year or so, APHIS did come out with a 330 rule. And rather than list all those biocontrol agents on the 330 rule, I'll just have you reference and Google that. And that is an entire list of biocontrol agents that you no longer need a permit for. They are considered ubiquitous and almost naturalized. So the paperwork for APHIS is going to be, hopefully for them, less and less in the future because they have that 330 rule. That's sort of a new thing. So you need to know what you need. Know what you need for biocontrol interstate movement. That's going to be the 526 form. Intrastate movement is actually left up to the states. So if you're going to move something within your respective state, you don't need a permit per se, but it is a good idea to involve APHIS and let them know what you're doing in case you're working with something that they don't really want you to work on, or something that might not be something that you should even touch or bother with. So I wanted to briefly mention what you do after you get that permit and you do the releases. And that's, we need to monitor. We need to know what's working and what's not. We've come up with a method to do that. That's the Standardized Impact Monitoring Protocol, or SIMP. And if you go to ibiocontrol.org, we have all of the information you need for SIMP on there. We currently have these systems that are biocontrol systems that we're working on. We've got pre-release for these guys, and we're kind of moving into operational with hoary crest and yellow star thistle starting next year with some new biocontrol agents that have recently been approved. And I wanted to real quickly talk about the International Code of Best Practices before I turn it over to John. I talked about everything that we have that really governs what biocontrol does from a legal standpoint. We as a biocontrol community like to go above and beyond that, and we adopted the International Code of Best Practices in 1999. This really makes sure that we kind of set the bar a little bit higher than what we're legally required to do, and in so doing, we make sure biocontrol is available to you guys going into the future. This is a science that could be easily derailed, and so I would encourage all of you that are involved in biocontrol to make sure you operate within the International Code of Best Practices. So I kind of want to leave you with this before John takes over: IPM and biocontrol. Biological control is an underutilized IPM tool. We in the BLM primarily work with herbicides. I think a lot of folks are in that same boat. The science behind biological control is very sound. Host specificity testing is very complete. Genetic work is a new wrinkle that we're throwing in that's been able to take away a lot of the guesswork. Know the objective, the tool you need for your objective. If you want to eradicate something, biocontrol is not going to work for you. Refer to that ideal biocontrol result slide. You're never going to get rid of the target weed, but what you can do is suppress it or contain it. Examine all possible methods. Look at mechanical, cultural, chemical. Figure out if biocontrol might work, and if it does, then use it as a tool. You've got a host of resources that John's going to talk about, and with that, I'll leave my contact information up here for a quick second, and then I'm going to stop sharing and turn it over to John.
Okay, thank you so much, Joey. That was fantastic. We're going to hold the questions until the end, but one question that was asked, Joey, is that you mentioned a cost-effective study. for biological control. And if there's a link you could put in the chat to that study, that would, folks are interested in that link.
Okay, I'll see what I can do.
Thank you so much. Okay, we're gonna switch it up here. And our next speaker is John Kaltenbach with the Colorado Department of Agriculture. He's the biological control specialist and has been with the Department of Agriculture for 12 years, the last 6 of which has been with the Palisade Insectary Biological Control Program. John is the Director of Front Range Operations for the Insectary and works mainly in the Front Range and Eastern Colorado areas. The Insectary imports, rears, establishes, and colonizes new beneficial organisms for the control of specific plant and insect pests, and also evaluates their effectiveness. John has a Bachelor's of Science in Entomology from the University of Wyoming and 3 years postgraduate education with the University of Arizona. He has over 30 years of experience working in entomology, including plant pest management, regulation and control with USDA, APHIS, PPQ, as an entomology research assistant at the University of Arizona and the University of Wyoming, 2 years as a Peace Corps volunteer promoting and implementing integrated pest management in Guatemala. John is a member of NASMA and a member of the Colorado Emerald Ash Borer Response Team. I'm so happy to have you with us today, John. Thank you so much, and I'll let you take it away.
Thanks, Elizabeth. I have already messed up. Thanks for the introduction, Elizabeth, and thanks, Joey, for the great beginning part of the talk. I'm going to cover some of the same stuff Joey covered, just to emphasize what's going on with biocontrol. And I'm not really going to talk about arthropod biocontrol too much. I'm mostly going to focus on weeds. I figured that was kind of the theme of what Joey was going to talk about. So I'm going to talk about mostly the Colorado Department of Agriculture's biocontrol program, give you an idea of what we do, how we do our program, and then a little bit of why you should use biocontrol, and then some resources for you. The Palisade Insectary, where I don't work out of there, I work on the Front Range, but this is where all my boss and all my colleagues work. The Palisade Insectary was started 75 years ago in the 1940s for oriental fruit moth control, and they found a parasitoid that killed oriental fruit moth. Unfortunately, it didn't overwinter, so they came up with a mass rearing method, and they've been doing that since the early 40s. We figure it saves peach growers in the Grand Valley 3 to 4 pesticide sprays every year, and you wouldn't be able to have organic peaches at all without this program. Currently, the Insectary works on over 20 biocontrol agents for the control of insects and weeds. We consider ourselves a partner in pest management. We work in the state, we work regionally, we work for the whole US, and some we collaborate internationally as well. And our main goal is to work with landowners and the general public to control invasive species. Palisade, just for reference, is on the west slope of Colorado over here. I live in the Denver area, and our headquarters office is in Broomfield. And you can see Colorado, we try and cover the whole state, so we've got quite a ways to drive if we want to get to the other side of the state, so it helps having me over here a little closer to some of the action. Again, we were really hoping to have a 75th anniversary party in September for the Insectary. Unfortunately, with COVID, we're not doing that. We'll have to come up with some other way to celebrate. We haven't figured that out yet, and maybe we'll just have to delay it until all this is over. We can get together and have a big party. This is sort of the stuff that Joey went over, the steps in weed biocontrol. He said 10 to 15 years to get approval. And so we were talking about identification of the target, overseas exploration, quarantine work, and that kind of stuff. Where we are, what I do in Colorado is some field testing and then the full-scale implementation. So we're all the way at the end of the process, getting it out into the field. We look at our program, kind of have 3 parts with our program. Implementation, and that can be rearing in the Insectary, or we do a lot of what we call field nursery collection. And the idea is to get those agents out into the field and get them working. We do monitoring. We track the biocontrol agents and the target pests and see how that's how effective that is. And then we also look for those non-target effects that we're worried about. And then, of course, we do a lot of education. Basically, you know, biocontrol, it is underutilized. A lot of people don't know about it. So we spend a lot of time educating people like, hey, this is something you can utilize. This is how to use it. And we do that through a lot of different means. So, so here's rearing mac at the insectary. It's a artificial, or it's a it's a alternate host rearing. It's quite a process. They just kind of keep the culture alive through the winter, and then in the spring they start building it up. And between 1.5 and 2 million wasps are released every summer, and basically just 1,000 pupa of the wasp. And farmers come by and pick up their wasps at the insectary, and then they go out and hang them in trees. On the right, you can see that's a collection of Canada thistle rust. That's a different one. It's not an insect herbivore. We mostly talked about insects, but we also have a rust fungus we're using for Canada thistle. And so we do some field collection of that. So we have field nurseries. We can also collect insects out in the field a couple different ways, but a lot of it's done with a sweep net. And then when you're implementing, you're going to go out and you're going to release the weevils or insects wherever you're at, your agents. And hopefully you're establishing insect nursery sites in the field, or maybe you're doing a release for those monitoring and research to gauge impact. And then on the right, we do ship a lot of insects. We collect, sort, and ship them to cooperators. And we have some customers too, which I'll talk a little bit about that project. For monitoring, you know, we want to know where the biocontrol agents are. We want to know if they're effective. We want to know if they're having an effect on the target species. We're going to look for non-target species as well. And we use all that information to inform pest management decisions. So on the left, this is the SIMP transect that Joey talked about. It's a 20-meter transect with a frame to monitor vegetation every 2 meters. That helps us identify how the target pest is doing, and whether the density is going down or not. I can tell you from having done this the last, I've been full-time the last 7 years, but I've been doing this work for the last 14 years. And your memory of what something looks like when you're out in the field can change over years. And having some hard data, some numbers, really helps to know what's going on out in the field, because sometimes you get out there and it's like, oh, it doesn't look any different, but then you actually do the counts and you see that stem density has gone down, or you see that the other ecological communities, some other things have come back, some woody species, some forbs, some perennial grasses, things like that. On the right, we have a dead tamarisk tree, which is always good to see. So tamarisk is a little different. It's not a transect monitoring. We're doing individual trees. We tag trees and then we monitor them year by year and monitor the effects of Diarrhea carinulata on tamarisk in Colorado. One of our other monitorings that we do is macro plot monitoring, which is a 16 square meter stem density monitoring around a release point. And that's on the left here. And each of these squares is a meter square. And we do a census of the target pest in there, or the target weed in there. And we look for our agent, and also look for flowering and non-flowering, some other impacts that maybe the agent has. And then on the right, this is, we do some 50-meter transects, 6 50-meter transects from each release point. And then every 2 meters, we monitor the ecological community. So that gives us an idea of the spread of the weeds, and also the we can measure the ecological community if the grass, the forbs, the weeds are there increasing or decreasing. These are some of the forms that we use for the macro plot monitoring. You can see there's a lot of data we collect for each site: non-flowering stems, flowering stems, the height of stems. And then over here, we're collecting whether toad flax is present or not. This is a particular one for toad flax, or whatever your target weed is. And then your community composition of, which is breaking out into grass, forbs, woody species, and also monitoring litter and bare soil. Here's the form for the standard impact monitoring protocol that Joey talked about. And all this is really important for us, because we want to take this information and we want to be able to help landowners and land managers. And if we don't have this info to say, hey, we know this works, then I'll say it really helps to have this info to convince folks to try biocontrol. Our education, we do a lot of different things for education. Of course, the Palisade Insectary has been working with peach farmers since 1945, but the end users need to know sort of about biocontrol and what they can expect from it. So that's a big part of our job. For the last, I don't know, 5 or 6 years, the Insectaries had a educational booth at the Palisade International Honeybee Festival. That was canceled this year, unfortunately. It was last weekend. But hopefully we'll get to do that again in the future. We do lots of fair work, educational talks, seminars, workshops, and then one of the real popular things is tours of the insectary, student groups, folks that work against noxious weeds and other folks. Of course, again, not going on this year. And then, of course, the usual websites, brochures, flyers. Pesticides definitely seem easier. Again, it's a good tool to have in your toolbox for integrated pest management. To implement the biology of the ecology of your system, we do that monitoring, understand things, and it's you want to know why. Basically, I screwed up. Basically, biological control is good to have as part of an integrated pest management approach, and it just depends on what your situation is and what your ultimate goal is. If your ultimate goal is to eradicate the invasive species, biocontrol is not going to be your preferred method. Maybe if it could be in a long stretch, if you don't have the money or the resources right now to control something, you can maybe implement some biocontrol with the thought in the future that you can eradicate it. If you and you can bring the density of your target pest down, you can keep it from spreading. Sometimes those are potential goals. But really, what you want to do with biocontrol is have a situation where if you can't use pesticides or it's too expensive or safe, it's effective, it can be very inexpensive and sustainable and keep going for you. Joey referred to some 9 knowledge we have of our agents. Effectiveness, success can be measured on a lot of different do it. In about 10% of cases, you get satisfactory control with, according to this paper that released in 2016, for about over 2,000 introductions. So you get some establishment at about a third of places, and then about 10% result in satisfactory control. And that doesn't seem like a lot, but when you consider those huge impacts, that cost-benefit ratio, that can be way over 1,000. I think Joey said 18,000 to 1 for one study. That can be really beneficial, really save you a lot of money, time, and that's what you'd like for biocontrol. And so for all the ones that don't work, where agents don't establish, because it's difficult to get things to establish sometimes, matching up climate and site characteristics for your agents with your target pests can be difficult. And so the successes that we've had far outweigh, or the benefits from those successes far outweigh the cost for the failures, for sure. Joey actually talked about this a little bit. St. John's wort. There's actually a monument in Humboldt, California, to the Chrysolina beetles that saved the rangeland up there. The sheep growers, the wool growers, put up this monument. That's one of my bucket list trips as a geeky entomologist. I want to go out there someday and see this. But he also showed these photos here, so I'll just skip through them real quick, the before and after photos. And in terms of monitoring, I should actually say this is really great. If you don't have time to do some of those monitoring things that I was talking about, where you're talking about getting down on your hands and knees and counting the density of plants, a good photo point can really tell a story, especially if you take the photo at peak flower and then go back every year and look at peak flower. For us in Colorado, some of the successes we've had, and it can be site dependent for sure. One is leafy spurge control. We've definitely had luck in a lot of places in Colorado where a mixture of Aphthona flea beetles and Oberia stem borers have taken care of leafy spurge for us. We do have some areas where it persists and maybe doesn't work as well. There are areas where the flea beetles might get flooded out, or the flea beetles might not overwinter well because it's a little too cold. They don't get snow cover over the top and cold snap, and they die off. But we do have customers anecdotally that tell us they release the flea beetles every year, and the years we don't provide them flea beetles, the leafy spurge comes back. And so that's an instance where it can work. It's not as long term as we'd hope, but it still works. And then one of our bigger successes in the last few years was a post-fire situation up outside of Fort Collins. We had a Hewlett Gulch and High Park fires burned a huge area of Colorado, and the area was just taken over by Dalmatian toadflax. You can see on the left, this is what it looked like the year after the fire, and there were just whole hillsides covered with the yellow Dalmatian toadflax. We released 8,000 weevils in an area that was maybe 1,000 acres of pretty rough terrain. I mean, this is pretty steep. There's actually steeper sections. It's not an area that we could cover with herbicides. You could put a backpack sprayer on somebody, but you'd have to hike up some pretty steep hills, and it wouldn't be very cost effective. We released 8,000 weevils, and over the course of about 3 or 4 years, the stem density, according to our monitoring, went from decreased by 99%. It's pretty amazing. And here's another shot of that. Here's what some of the toadflax looked like on the left. This is where releasing some weevils in 2013, and then here's the right. There's not any yellow toadflax that can be found in those areas, so it was a really good success for us. This is the macro plot monitoring we did out at that site, and I wanted to show you this graph specifically because this is the sort of data that you can get from the macro plot monitoring. You can see over here to the left, the Dalmatian toadflax density went way down, and you can see some other things like the forb count went up, and the woody species is up, and the leaf litter after the fire, there was no leaf litter on the ground at all, and that's really built up too. So, and just some other before and after. Here's some after shots. This is tamarisk mortality in Humboldt, and you can see mortality percentage of tamarisk going from biocontrols can be really good. Tamarisk biocontrol is one of our pretty successful agent in Colorado. It's what Joey was talking about in terms of not being able to move things interstate. Diarrhea carinulata is one of those that the USDA does not permit to move between state lines, but the beetles came into Colorado and about 15 years ago, and we've been able to collect and re-release them in Colorado. We made the decision to move them around Colorado ourselves, and we've had a lot of success with the beetles on the west slope, not so much on the east slope so far. And then here's some monitoring photos just to show you sort of how good a photo can demonstrate effectiveness of a biocontrol agent. So you get some fully leafed out tamarisk on the left, and a couple of years later, totally defoliated. And then here's some mortality counts. So how am I doing on time? I think I got about 5 minutes. And then just a few other things where about tamarisk and Diarrhea specifically is tree death not necessarily isn't going to necessarily happen, but canopy dieback is really important. Basically, the ecosystem can recover a little bit if you get rid of some of the canopy. Some of the other things can grow underneath it, and beetles can really help with saving water in our western waterways as well. So One of the things I really kind of wanted to highlight was one of our newer things that we've been working on the last few years, and it's not an insect that feeds on weeds. It's a rust fungus, Puccinia punctiformis. This is actually doesn't require didn't require the overseas exploration because we found rust fungus already in Colorado, and you can probably find it in your state too. It seems like the rust fungus gets around pretty well. And what you need to do is just go to a Canada thistle patch in the spring and look for sort of yellowish plants that have these yellow orangish pustules on the bottom, and they're spermagonia, and they smell really flowery and they're really sweet smelling. And that's your Canada thistle rust fungus. And we've been able to find it at about more than 50 places throughout Colorado. I think other folks around the western region have found it in places. Basically, this rust fungus has a complicated life cycle. In the early spring, you'd see plants like this. And I encourage you to go out into a Canada thistle patch and look for something like that. They're systemically diseased roots with Puccinia punctiformis, which is a rust, it's a root parasite. And then it hijacks the plant and it sends up systemically infected plants. And those are seen here on the right. In the summer, those spermagonia sexually cross and become aciospores. And then the fall, the spores fall on newly emerging rosettes and produce iridinia, which turn into teleospores, which eventually turn into basidiospores that grow hyphae back down into the roots. That's the life cycle of the Canada thistle rust fungus. And what we figured out is that we can harvest those teleospores and put them out in the fall. And we've seen some really dramatic results. Some of our transects with 100% decline of Canada thistle, 99% decline, on average about a 70% decline after 3 years with the Canada thistle rust fungus. So that's been really successful. And for Colorado specifically, we have what we call a request a bug program. So this is available to landowners, land managers in Colorado. They here's our agents that we're working with, very similar to the list that Joey showed of things that he's working on. So we have agents for Canada thistle, Dalmatian toadflax, diffuse knapweed, field bindweed, leafy spurge, musk thistle, puncture vine, Russian knapweed, spotted knapweed, and yellow toadflax. And we do charge money for those to recover some of our cost of getting those. And that's how we promote biocontrol around Colorado. Unfortunately, we can't ship out of state for this. If you need to get things in your state, you need to probably start with your state Department of Agriculture is a good place, or your county weed managers if you have such a thing in your state. Again, USDA APHIS PPQ is a great place. They're, of course, the ones with the 526 permits for interstate movement. And there's the best place there to get a hold of them would be the state plant health director's office. Every state has one. You can find their number online pretty easily. Give them a call if you're interested in some what's available for agents for what you have. Your state natural resource department as well also participates in biocontrol in a lot of instances, depends on where you are. And then, of course, a lot of help from university researchers, mostly land grant, but not necessarily just land grant, but a lot of university researchers also are good sources for helping you with biocontrol. And Joey mentioned this website, and I encourage you to go to it, ibiocontrol.org. Lots of great information on how to structure a biocontrol program. There's a catalog on there. I call it the Julian catalog, this biocontrol of weeds. You can download a copy of it. You can search the database. If you have a weed that you're interested in, look into that database, see what's been out there, see if something has already been tried. And then there's a lot of good information. And then additionally, they have an app that you can use when you go to release agents, to map agents, to track things. It's a really good website, and I can't promote that enough. And I got to thank everybody at the Palisade Insectary, specifically my boss, Dan Bean, and all my colleagues. We've had, for our program too, lots of seasonal and technical help over the years, technician help, and then tons of cooperators and collaborators. We don't do it alone. We do it with a lot of other people. So thank you. And there's my email address if you have any questions. And I will stop sharing my screen here in a minute and let other folks join in.
Okay, thank you so much, John. That was fantastic. And Joey has been in the Q&A answering questions, so I really appreciate that. Thank you so much, because we are a little short on time for questions. So I appreciate Joey getting in there and answering a lot of those questions for folks. And of course, as the presentation's gone on, you just naturally answered a lot of the questions as well. So a couple of the questions that are still open and I think, boy, Joey's just cranking through these, so this is awesome. A link to the rust control research, if you have that available, if you could put that in the chat, John.
Oh, link for rust control research. What would be the best place?
And there's compliments coming in to thank you for your excellent presentations today. So.
What is the estimated life of biocontrol? Well, some are like the St. John's wort out in California. I don't know if anybody still works on it. I think it's controlling St. John's wort to this day. That's the ideal goal, is when you release something and they do all the work. And actually, I would advocate that for a lot of biocontrol. You don't want to put a lot of work into it. Release them, let them do the reproduction and spread on their own. That's the ultimate success. So and then if your target pest dies out, then your agent dies out too. I don't think we have any evidence of host switching or anything in biocontrol. Pretty much when the host dies, the agent dies. I will see if, I don't know if I have a good link for rust. I'll have to, I don't know. Do you send things out later?
Yeah, absolutely. Yep, that's okay. Mm-hmm. I'm getting several folks asking if you could reshare both of your contact information. So if you have those available.
Yeah, Elizabeth, I put mine in the chat. So if somebody wants to look and see that, rather than share my screen again.
Yeah, perfect.
I can hear too if that works.
I would assume this is dependent on the species, but one of the questions in here is, what is the estimated life of biocontrol?
I did answer that one.
You did?
Yeah.
Okay.
Oh, no, I'm actually, I answered one like it. I'm sorry.
No, that's
yeah.
okay.
The estimated life cycle, typically these are univoltine, so 1 generation per year. Some of them are multivoltine, 2 or 3 generations per year. Some of them can do up to 10 to 15 generations, like Yap Yellow for Russian knapweed. So it really depends on what you're working with. John mentioned iBiocontrol. We're really, that's really been upgraded quite a bit. And I would encourage folks to go look there. I think most of the questions that have been in this pop-up chat have been addressed by iBiocontrol in one way or another. It's kind of the clearinghouse for all things biocontrol at this point. I don't Jeff Morissette's question about how long does SIMP stay in place after biocontrol is released has been answered. It really kind of, the SIMP is just a way to monitor, and it's as long as you want to keep it there is, I guess, the best answer for that. We have several sites that we've been going back to for 15 years at this point. The more data you have, the better. SIMP is designed to take anywhere from 30 to 45 minutes one time per year. So it's really not a huge investment of time. The power, the statistical power with SIMP is not in going out there in 2020, but it's in looking at every site from 2006 to now. And you can really, there's built-in tools within iBiocontrol and SIMP to kind of look at how those work. And yeah, it's been a very effective way to look at how biocontrol is doing in the field.
Okay. All right, well, thank you so much. We are just about out of time. And so, of course, we'll try to get through the last couple of questions in the Q&A and send some written responses out.
I put a link to ResearchGate info for Dr. Dana Berner. He's the main pathologist that did most of the rust fungus research. If you look at a couple of his papers, he shows really good information on Puccinia punctiformis. And then somebody asked the last 2 insects that were my slideshow. One was Siphocoleonis acades, which is the root weevil of knapweed. Works great. It's a big pinto bean looking weevil. Love that thing. The other one was just an awesome praying mantid that I found out in the field when I was sweeping. It was actually on me. So not a biocontrol agent we collect pre-release, just one of those natural predators out there that is doing a lot of work. So I just like the insect. So